Thank you for the nice welcome. Very happy to be here. From Pixelgen, we also have another colleague, Katie Healy, down there waving her hand. So if you want to chat with us after this short introduction to what we're up to at Pixelgen, we'd be happy to talk to you guys. And what we're building at Pixelgen is a new research area of protein interactomics. And we're doing this with a new chemistry that we developed that we're calling Proximity Networks, or the Proximity Network Assay. And if you think about cells and cell functions, those are governed by proteins, and proteins never really work alone. They work by interacting with other proteins. So cellular functions are executed actually by protein interactions. And this is vastly understudied in clinical research, especially, where you're studying real human samples. So if you kind of take a step back and look at the bigger picture, you have DNA or genomics, which is of course a blueprint, and RNA that's generated from the DNA, which most people study in single-cell RNA sequencing. This is a manufacturing message to make proteins that carry out the functions. And that is of course the third omics. But what about the regulation of functions beyond just abundance? Because the detection methods that we're using today, like single-cell RNA sequencing, flow cytometry, essentially they generate a parts list of which proteins or which RNAs are inside the cell. But the protein interactions goes beyond the parts list and looks at the functional organization of protein assemblies. And this is the ultimate regulator of cell functions. When cells migrate, when they interact with other cells, when they are about to go into a tissue or a tumor, and so on. So all these essential functions of cells are driven by protein interactions, and especially so on the surface of cells, not just intracellular protein interactions, but also the organization on the surface. And comparing various technologies for analyzing the proteomic information of single cells. You have, of course, your CytSeq, which can generate a very large parts list of proteins on cells. Mass cytometry and flow cytometry, everyone is familiar with. You can assay thousands or hundreds of thousands of cells with parts lists of proteins. But if you want the spatial resolution, you have to go into the microscopy dimension with either medium resolution microscopy with 2 or 3 colors, or super resolution microscopy with either lower resolution. And these technologies don't really scale. We've been very fortunate to have genomics, proteomics, and transcriptomics technologies become very powerful to generate vast amounts of data. But what about protein interactions on single cells? There's no such technique. So the Proximity Network Assay delivers this quadrant here with 155 proteins that we are detecting with antibodies at 50 nanometers spatial resolution in 3-dimensional data. So this is actual real data that's generated from the method and as projected by the software. So here you get 155 surface proteins located at 50,000 spatial locations around a T cell, B cell, or whatever type of cell you're analyzing in a heterogeneous sample. And you can assay all these markers simultaneously in 1,000 cells in about 8 samples. And this is a very straightforward analysis procedure. So how does that really work? So a Proximity Network Assay, you take cells in suspension, which could be from PBMC sample, cryopreserved samples, bone marrow samples, cell lines, or dissociated fresh human tissues. These are fixed lightly with PFA to lock all the protein positions in place. And then we have antibodies with DNA tags, very similar to what we developed at Olink, where we gained scalability in protein detection in plasma. But here, this is of course for protein interaction detection. So each antibody then binds to the recombinant antigen target, and the DNA tags on the antibodies are then amplified briefly with a rolling circle replication. So this makes multiple copies of the tag on the antibody. And once you have multiple copies on the tag, you can make multiple connections to the nearby antibody DNA tags. So very similar to proximity ligation or proximity extension, But here we're calling this Proximity Network Assay because you can make multiple connections, which forms a network that sprawls and encompasses entire single cells. So at the end of the reaction, you get a PCR product that you feed your next-gen sequencer, Illumina, MGI, Ultima, Element, any sequencer that you may have. And then there's a software at the end of the reaction that analyzes the spatial positions. So this is kind of illustrated here in this slide. The input is single cells or cell-cell complexes, but always in a suspension. And since this is a chemistry assay, it's not dependent on light. You get a three-dimensional image that looks at the cell from all angles at the same time, right? And the kit that we've built that enables this is very simple to use. You can run it in any lab. All you need is a thermocycler and a centrifuge. Then you can pass your PCR product to your sequencing core facility, and then the software figures out where all the proteins are on these thousands of cells and generates these three-dimensional reconstructions of the single cells or cell-cell complexes. So it's very, very easy to use. And the data that we get out of this is, of course, this spatial network of where the proteins are for each single cell. And we naturally get also the abundance of each protein with this method, just like you would do with CiteSeq or flow cytometry. So if you have a low amount or a high amount, and then we've developed something that we call the clustering metric. So if one protein is randomly dispersed across the entire cell, or if it's clustered in islands, the clustering score would then go up. So, and then the third metric is the colocalization metric, and this is where we're getting into the protein interactomics, where if two proteins are colocalized within one distance in the graph network more often than predicted by random chance, random abundance chance, then this colocalization score becomes positive. If it's random, it's around 0. And if it's segregated, it becomes negative. And segregations of proteins on the surface of immune cells is very common to regulate migration, to have a front and a back end of cells. We see this a lot in the data set together with our clients, as well as various adhesion markers that would cluster when a cell is about to attach somewhere. So there's a lot of information on the spatial organization that you would miss if you only make these parts lists with flow cytometry or CiteSeq. There's a preprint out on this technology where you can read up on the method in more detail. It's under review in Nature Methods. Here you can study the validation. All the antibodies are highly validated with flow cytometry. We validated the spatial scores with microscopy. So this is microscopy for CD81, for example. You can see how it's, maybe I'm hiding it. So it's kind of spotted like this. So CD81 is rather clustered while CD40 is not. And then that shows up in our data as a high proximity score when it's clustered, or low if it's not. And then you can plot the cells and you can see these clusters here in our data with the proximity network, or if it's dispersed. And you can generate these types of plots. And this is highly useful as a new dimension of biomarkers, right? So we've been studying these parts list methods as new biomarkers or drug targets, but protein interactomics then is a possibility to develop new biomarkers for autoimmune disease, leukemias, lymphomas, and so on, since we are developing this method in that area with those types of antibodies against immune cell surface proteins. So this can be used for disease profiling and figuring out what are the molecular mechanisms behind various cell functions, or when cell functions go wrong, what's happening and what can we possibly do about it when we understand this. You can identify potential new drug targets. So there's about 1,000 proteins on the surface of a cell, but if you start looking at protein interactions, you have 1,000 times 1,000 potential interactions at the maximum theoretical amount, which then generates a new druggable space on the surface of cells. And 60% of all drugs target proteins on the surface of cells, especially in immuno-oncology. So we've done a bunch of studies with this method to highlight its applicability, especially in CAR T cells. So the CAR T community is very interested in understanding the spatial organization of the CAR on their CAR T cells. And you can use this method then to dissect this CAR T cell membrane interactome and compare various different types of CAR T constructs. So from our data, you would, in a CAR T cell that has been put into a patient or is just assayed in vitro in a PBMC cell dataset, you get all your typical cell annotations based on abundance, so you can find your monocytes, NK cells, and so on in the datasets. But the CAR T cells will then pop out as having the CAR. So we have a bunch of various extra probes that you can add to your 155. So like an FMC63 probe, G4S probe, which is the typical linker that goes between the CAR, flag tag linkers and so on. You can kind of see this as a toolbox of various things that you can add. And this particular CAR T cell has a very evenly dispersed CAR. And then you can study the CD19 CAR T cell interactome, which proteins are near it. So, for example, if you have certain proteins very near the CAR, it can regulate or upregulate or deregulate the activity. So what we've seen in most of our data sets is that the CD19 CAR is localized very similar to the T cell receptors, which sounds, at least to me, like a very positive thing. It's next to the TCR-AB and other proteins that are in the vicinity typically of the T cell receptor. You can do a lot of in vitro co-cultures where you take your CAR T cells and culture them with a cancer cell, like a cancer cell line. And with this co-culture over time, you get a lot of abundance changes, of course, because the CAR T cells get activated, etc. And that can be studied easily with this method. But it gets a little more interesting once we start studying the spatial organization. Because when assaying the function of these CD19 chimeric antigen receptors in the co-cultures, what happens is that a bunch of cancer B cell markers the cancer cell proteins start emerging on the CAR T cells. And you can see that in abundance. So B cell markers are then appearing on the CAR T cell, but they're always very spotted and patchy. And this is because what's happening is the trogocytosis process, where CAR T cells are binding to tumor cells, and you get a patch of tumor cell material on the CAR T cell. And this can then be analyzed with our software. How many patches do you have per cell? Because this is a spatial organization problem, right? So you can have no 1, 2, or 3 patches, and you can start counting them and associating them with various potential exhaustion and activation markers like CD25 and PD-1. You can also look at the cell-cell complexes that appear and quantify them. So if you have a Raji cell and the CAR T cell, you have CD2 on one half of the graph network and CD40 on the other half. So that Raji cells alone, of course, have CD20, T cells alone will have CD2. And in the co-culture, you start getting these complexes, then eventually disappear because the cancer cells are getting killed. And you can quantify them very easily because the data here picks up these cell-cell complexes very well because it's a segregation of B cell markers from T cell markers. So other applications of this technology is to use it as this new potential biomarker realm, as I mentioned. So one problem that we've studied together with Aaron Vinny at Columbia is to look at responders to AML treatment, so acute myeloid leukemia. So these patients are treated with hypomethylating agents, and only about 20% of them respond, but they respond really well. So is there any feature of those patients that we could see? So we did a smaller pilot study together with them with a number of responding patients and non-responding and put that through a random forest classifier. And the amount of blasts prior to treatment wasn't really indicative of response to this treatment. But once we started building a total score of abundance, clustering, and co-localization metrics, we got an accuracy of about 94% in predicting responders versus non-responders. So this is just a small pilot, but it kind of gives you an idea of where this type of technology could potentially be heading in helping treat the right patients with the right treatments. As these protein interactions are hugely important for cellular functions. We have lots and lots of more application examples like this from clients all around the world that's using this technology. And just a little thing more about Pixelgen as a company. We've been around for about 4 or 5 years. We're located in Solna, and we have about 40 people working all the way from production to sales and marketing and support and so on. So we're looking forward to working with more clients here in Huddinge. We have a few already. So applications where this is taking off mostly is in hematology, in blood cancers. There's a lot of mystery still remaining there to figure out in these types of diseases. Immune therapy and cell engineering, like the CAR-T data that I showed. A lot of interest there. And who will benefit from CAR-T therapy, who might potentially have an adverse event with CAR-T therapy. There's lots of very interesting questions that you can ask there. And basic immunology, just understanding immune cell functions, autoimmune disease, and also in drug development. A few pharma companies are already using this method as well. And as I mentioned initially, it's a very simple kit-based product. So imagine assaying 155 proteins and their potential protein interactions, so 155 times 155 with a microscope in thousands of cells in 3D. It's just impossible. But we can accomplish this with this simple kit that you can run in any of your labs here. And we validated the data, of course, by making sure that in the data we actually find the known interactions, protein interactions. So if I go back a little bit here, maybe I jumped that a bit too quickly, that looking at known protein complexes like the MHC1, which is HLA-ABC, and the beta-2 microglobulin complex, all of those types of well-known complexes emerge in our data sets at the single cell level. LFA1, B-cell receptor complexes, and so on. So that kind of validates and assures us that this method actually detects these protein interactions. So once you find new novel ones, you can be certain that they are true, and you can validate them one by one with microscopy as well. So I think I'm going to stop there and hopefully get a few of your questions. I'm looking forward to hearing your thoughts on this.